Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
What if the key to fighting Alzheimer's disease isn't just about, clearing plaques, but about preserving your brain's internal operating system? Today on the Deep Dive, we're unpacking a really groundbreaking new study. It reveals something quite remarkable. As Alzheimer's progresses, our brain cells seem to experience this widespread breakdown in their epigenomic information.
Right, think of it like the instruction manual getting corrupted.
Exactly. The instructions telling genes what to do, when to do it. But here's the really powerful twist. Individuals who keep their cognitive abilities sharp, even with significant Alzheimer's pathology, they actually preserve this epigenomic stability.
Which is fascinating.
It really is. This isn't just some minor detail. It suggests a whole new way to look at understanding and maybe even treating the disease.
It's a completely new angle.
So we're diving deep into a remarkable 2025 Cell study by Liu, Zhang, James, and their colleagues. Our mission today, to kind of cut through the complexity of this incredibly detailed map they've made of brain changes, we want to give you a clear understanding of this new frontier in Alzheimer's research, and also this idea of cognitive resilience.
Yeah, and to really get started, it's important to remember that Alzheimer's doesn't hit the brain all at once.
Yeah.
It's selective. Some regions, unfortunately, decline quite early on. Others manage to stay relatively okay for longer.
And then there's that resilience factor.
Exactly, cognitive resilience. These are the people who stay incredibly sharp mentally, even when, if you looked at their brains, you'd see significant signs of Alzheimer's pathology. It's quite extraordinary.
How did older methods miss this?
Well, for a long time, the traditional research methods, they often missed this crucial variability because they basically blended all the cells together in a sample, like making a smoothie out of the brain tissue.
Losing the details.
Precisely. More recent single-cell RNA studies, they were revolutionary. They started showing us which genes are turning on or off in individual cells. Huge step forward. But even those studies don't fully capture the underlying chromatin programs. These are the fundamental structures deep within your DNA that actually control whether those genes can even be accessed to be turned on or off.
Okay, so that's where epigenomics comes in.
That's exactly it. The big knowledge gap, the missing piece, has been getting a really detailed, region-aware, and cell-type-specific map of these chromatin changes across the whole spectrum of Alzheimer's, from early to late stages, and crucially, looking specifically at those resilient individuals.
So this study is really trying to fill that gap.
It absolutely pushes the boundaries to do just that.
Okay, let's unpack how they built this incredibly detailed map then. The researchers didn't just glance at one part of the brain or a few cells, did they? It sounds like a massive undertaking.
Oh, it was huge. They examined the brains of individuals at all different stages of Alzheimer's. What did this really comprehensive approach tell them about the brain's cellular landscape?
What was the scale?
The scale is, well, it's truly impressive, almost like building a Google Maps of the Alzheimer's brain, but at the cellular level. They meticulously analyzed 384 post-mortem brain samples. These came from 111 different individuals, carefully categorized as normal, early-stage AD, and late-stage AD. So you get the full picture, the whole progression.
And multiple regions.
Yes, not just one area. They looked at 6 critical brain regions. And what's important here is that these regions have different vulnerabilities to AD, like the entorhinal cortex and hippocampus.
Yeah.
Those tend to be hit earliest and hardest.
And the techniques, you mentioned epigenomics.
Right, they use really cutting-edge techniques, things like single-nucleus ATAC-seq that tells you which parts of the DNA are physically accessible, kind of open for business, and also single-nucleus RNA-seq, which shows which genes are actually being read out, being expressed.
And they combine these.
Yes, they also use something called multi-ohm data sets. This is super powerful because it lets you get both kinds of data, the epigenomic accessibility and the gene expression, from the very same individual cell.
Wow, from one cell.
Yeah. It allowed them to profile over 3.5 million nuclei, just an enormous amount of data. And from all that, they built this incredibly granular classification, identifying 7 major cell classes, then 34 subclasses, and even drilling down to 67 high-resolution cell subtypes.
That level of detail seems crucial.
It was absolutely key, essential for pinpointing the really subtle changes happening in Alzheimer's disease.
So, okay, they've got this incredibly detailed cellular atlas. What did they actually discover about how these different cell types behave in the different brain regions they looked at?
Well, what they found really underscores the incredible diversity. The
Yeah.
brain isn't uniform at all. For instance, the neocortical regions like the angular gyrus or the prefrontal cortex, they tend to have a higher proportion of excitatory neurons. Makes sense. But the entorhinal cortex and hippocampus, those regions that are super vulnerable early on, they contain their own unique subtypes of excitatory neurons. Different flavors, if you will.
And the thalamus, you mentioned that was distinct.
Yeah, the thalamus, a region deeper in the brain, was particularly unique. It showed fewer neurons overall, but a higher proportion of glial cells, the support cells and vascular cells, compared to the other regions they studied.
Interesting. What about the regulators, the transcription factors?
Ah, yes. This is fascinating. When they look closely at the master regulators, the transcription factors specific to these cell subtypes, they discovered unique ones in the thalamic neurons. These included things called core circadian regulators like NR1D12 and RORs. These are genes involved in our internal clocks.
Circadian, like sleep cycles?
Exactly. And if you connect this to the bigger picture, these specific transcription factors are known to be involved in pathways related to, well, neuronal development, synaptic signaling, definitely sleep regulation, but also memory and cognition.
Which fits perfectly with what the thalamus does.
It aligns perfectly. The thalamus plays a crucial role in sleep-wake cycles, alertness, relaying sensory info, and cognitive functions. So this finding provides a really tangible molecular link between specific epigenomic regulators, these circadian factors, and essential brain functions known to be associated with the thalamus.
That's a great connection. Okay, so they've mapped the cells, the regions, the master regulators, but how do these regulators, these transcription factors, actually control the genes? What are the specific switches they were looking at?
Right, the actual control mechanisms. That's where the rubber meets the road, epigenomically speaking. The researchers identified over 1 million candidate cis-regulatory elements, or CCREs. Yeah. A million. You can think of these CCREs as individual control panels, or dimmer switches, scattered across our DNA. They dictate when, where, and how strongly genes get turned on or off. And what's more, they found these CCREs often don't work in isolation. They organize them into 123 cis-regulatory modules.
Modules, like groups of switches working together.
Exactly like that. Groups of these control panels that coordinate their activity, often in a very cell-type specific way. It shows this beautiful coordinated regulation. And importantly, they found a strong agreement between the activity of these epigenomic switches, the CCREs, and the actual gene expression levels they saw from the RNA data. That really validated their findings, showing the link was real.
Okay, makes sense. And then connecting these tiny genetic switches to the bigger picture of human diseases like Alzheimer's. Did the study shed light on the genetic risks we hear about?
Absolutely. They did a really clever thing. They leveraged data from genome-wide association studies, GWAS data. This data tells us about genetic variants across the population that are associated with a higher risk for certain diseases. They looked at where these known risk variants fall within their map of regulatory elements. And a really striking finding emerged for Alzheimer's.
What was it?
The Alzheimer's disease risk variants were highly enriched, meaning they clustered significantly in the CCREs, active specifically in microglia or other immune cells in the brain.
Microglia, the brain's immune cells.
Precisely.
Yeah.
This strongly aligns with and adds huge support to existing theories about the critical role of the brain's immune system, particularly microglia, in driving Alzheimer's progression. It really solidifies the idea that immune dysfunction in the brain is a major player.
Does it confirm that immune connection?
It provides very strong evidence for it at the regulatory level, yes. They also noted something interesting for other conditions. Variants linked to things like bipolar disorder, intelligence, schizophrenia, depression, those were enriched in neuron CCREs.
So different diseases linked to different cell types regulation.
Exactly. It really emphasizes the cell-type specific nature of disease risk throughout the brain. It underscores how targeted our understanding and potentially our treatments need to be.
Right. Okay, now let's get into the what's going wrong part in Alzheimer's at this fundamental level. The paper uses terms like epigenomic compartments and compromised compartmentalization. What exactly does that mean? Why is it so important?
Yeah, this is a key concept. Our genome, our DNA, isn't just a long string floating randomly in the nucleus. It's meticulously organized. Think of it like a very carefully organized digital library. You have distinct active compartments. These are regions of DNA that are open, accessible, like books readily available on open shelves. Genes here can be easily read. And then you have repressive compartments, regions that are tightly packed, less accessible, like books stored away in restricted archives. Genes here are generally kept quiet.
Okay, active and repressed zones.
Exactly. Now, what this study found in Alzheimer's is something they called globally compromised compartmentalization. It's like that carefully organized library system completely breaks down. The organization falls apart.
How so?
Well, active regions that should be open and accessible suddenly become hidden away, repressed. And conversely, normally hidden, repressed regions become exposed, activated inappropriately. It's a mess.
So the whole organization gets scrambled.
Profoundly scrambled. It's not just a few genes misbehaving. It's a large-scale epigenomic disarray across the entire genome. For instance, they found specific examples, typically repressive chromosomes, like chromosome 18, which is normally tucked away near the edge of the nucleus. It becomes more activated, more open in late-stage AD. And the opposite happened for active chromosomes, like chromosome 19, which is usually centrally located and accessible, becomes more repressed, less open. This isn't just a minor tweak. It's a major, widespread breakdown in the fundamental organization of the genome.
Wow. So this isn't just a slight shift, but a major, widespread breakdown in the brain's filing system, as you put it. What are the actual consequences of this breakdown for how genes get expressed?
The consequences are, well, significant and far-reaching. This epigenomic erosion, this breakdown in organization, leads directly to what they termed a global decline in transcriptomic fidelity.
Transcriptomic fidelity, meaning?
Meaning, in simple terms, the genes aren't being expressed correctly anymore, the accuracy is lost, genes that should be silent get inappropriately switched on, genes that should be active get switched off.
So the cell's instruction manual is basically becoming corrupted.
That's a perfect analogy. It's becoming corrupted, leading to widespread cellular dysfunction because the cells aren't getting the right instructions. And, perhaps not surprisingly, the most vulnerable brain regions, the entorhinal cortex and hippocampus, showed the strongest signs of these epigenomic alterations, this breakdown.
The early hit regions again.
Exactly. And within those regions, the problem was particularly pronounced in certain cell types. Oligodendrocytes, which make myelin, their precursors called OPCs, and microglia and other immune cells. This pattern perfectly aligns with how Alzheimer's pathology is known to progress through the brain, typically affecting these specific regions and cell types earliest and most severely.
Okay, that brings us to this concept of epigenomic information. It sounds really intriguing, almost like cells are literally losing information, losing their instruction manual as Alzheimer's progresses. Can you elaborate on what this information loss really means? Where is it most apparent?
Yes, it's a really powerful concept. It draws from something called the information and erosion theory of aging. The core idea here is that epigenomic information, which is essentially the set of instructions that dictates the cell's identity and function, what makes a neuron or a glial cell, this information increases during development as cells specialize. But, the theory goes, this information is progressively lost during aging, and especially in diseases like Alzheimer's.
So how do they measure this information?
They developed a way to quantify it. Essentially, by looking at how well the cells were maintaining their critical regulatory signals within those CCREs we talked about. A higher score meant the cell was holding onto its identity signals better. And crucially, what they observed was a global reduction in this epigenomic information in late-stage Alzheimer's. It was particularly bad in those highly vulnerable regions again, the entorhinal cortex and hippocampus.
Was it happening in all cells equally?
No, it wasn't uniform. They could pinpoint specific vulnerable cell types where this loss was most notable. Glial cells showed significant loss. That's your oligodendrocytes, OPCs, and microglia. But also certain types of excitatory neurons in the entorhinal cortex and hippocampus, and even a particular subtype of inhibitory neuron. So it really pinpoints where this cellular instruction manual is getting most severely degraded.
That suggests the glial cells, the brain support system, are really taking a hit. How do these epigenomic changes in glia relate to what they're supposed to do in the disease? Especially when they try to become activated to help out.
This is where it gets really interesting and actually quite tragic for the glial cells. Initially, when glial cells like microglia and astrocytes sense trouble, like the buildup of plaques, they actually increase their epigenomic information.
They increase it.
Yes, initially. It's like a necessary reprogramming. They need to change their state to become reactive, to try and fight the disease, clear debris, or initiate repair. It's an adaptive, probably helpful response at first.
Okay.
But the study found that with sustained activation, like you see in late-stage Alzheimer's where the pathology is chronic, these very same glial cells experience a substantial decline in their epigenomic information. They plummet. This leads to what the researchers called an exhausted state. It's like they completely burn out from constantly trying to fight the disease. They lose their proper functional identity in the process.
So they try to help, but eventually just collapse functionally.
That seems to be the story, yes. And this exhaustion is critical because healthy glial cells are absolutely essential for supporting neurons and maintaining brain health. And what's more, they connected this glial exhaustion directly to Alzheimer's genetic risk.
How so?
Individuals carrying the APOE4 gene variant, which is the major genetic risk factor for late-onset Alzheimer's, they showed the most severe epigenomic erosion, specifically in their glial cells.
Wow. So the gene risk makes the support cells more vulnerable to burnout.
It appears so. It's a tangible link. Genetic susceptibility seems to directly accelerate this epigenomic instability and ultimately lead to more severe glial dysfunction in the context of the disease. It's like having APOE4 means your cellular support crew burns out faster when faced with Alzheimer's pathology.
Okay, that's quite bleak, but this is where the story takes a more hopeful turn, right? The study also looked at those remarkable people with cognitive resilience, the ones whose brains seem to be fighting back effectively. What do they find in their epigenomes?
Yes, this is truly the breakthrough finding and the hopeful part. While Alzheimer's progression was clearly linked to this global loss of epigenomic information, cognitively resilient individuals show the exact opposite, a global increase in epigenomic information across their brain cells.
An increase, so they weren't just losing less, they were actively higher?
Exactly. It wasn't just preservation, it was enhancement in some ways. A direct contrast, suggesting a really powerful protective mechanism is at play in these individuals. And get this, the very same vulnerable cell types that showed the most dramatic information loss in AD patients. Remember, the EC and HCC excitatory neurons, specific inhibitory neurons, those inflammatory oligodendrocytes.
Yeah.
In the resilient individuals, those exact same cell types showed increases in epigenomic information. They were holding onto their identity, their instruction manual, much better. And this preservation also extended to the activated glial cells in resilient individuals. It seems to protect them from falling into that detrimental, exhausted state we saw in the AD patients.
That's incredible. So it's not just that they're coping better symptomatically, their actual cells at this fundamental epigenomic level are actively maintaining a healthier, more organized state. What does this mean for the sheer number of these crucial cells? Does stability keep them alive?
It seems to, yes. This epigenomic stability has direct implications for cell survival and population numbers. The study found a really clear correlation. Higher epigenomic information scores were strongly linked to the preservation of crucial cell types, especially those vulnerable excitatory and inhibitory neurons. More information meant more cells sticking around. Conversely, lower epigenomic information, that loss we saw in AD, was directly tied to the depletion of these neurons as the disease progressed.
So maintaining the instructions keeps the cells themselves from disappearing.
That's what the data strongly suggests. It highlights a fundamental truth here. Maintaining epigenomic integrity, keeping that cellular instruction manual intact, seems crucial for keeping important cell populations alive and functioning. It offers a clear potential mechanism for protection against the devastating cell loss seen in Alzheimer's.
Okay, let's pull back the curtain a bit more on the molecular machinery. What specific genes and pathways did they find that are involved in either maintaining this crucial epigenomic stability in resilient folks or contributing to its breakdown in AD?
Right, they dug into the specific molecular players. For epigenomic stability, they looked for genes whose expression levels went up along with higher epigenomic information scores. They found genes like DSCAM, ASTN2, and VCAN. Now, what's really telling is that these particular genes are known to be involved in pathways related to nuclear and chromatin structure organization.
So genes involved in maintaining the physical structure of the genome itself.
Exactly. It strongly suggests that actively maintaining the cell's internal architecture, its physical blueprint, is absolutely key for keeping the epigenome healthy and stable, like reinforcing the library building helps keep the books organized. And interestingly, they also found some known ADGUS genes previously linked to Alzheimer's risk among these potentially protective stability-associated genes like DNND4A and ENSA, which is complex but interesting.
Okay, and what about the flip side? the genes associated with the breakdown, the erosion.
On that side, they looked for genes whose expression went up as epigenomic information went down. These included genes like CST3, EGLN3, MT3, and significantly APOE, that major Alzheimer's risk factor gene, was strongly associated with erosion, especially in astrocytes, a type of glial cell.
And what did these genes do?
These erosion genes are heavily involved in pathways related to neurodegeneration, inflammation, and cellular stress responses, things like programmed cell death, apoptosis, and oxidative stress.
So as the epigenome breaks down, genes linked to damage and inflammation ramp up.
Precisely. It suggests a damaging feedback loop. Epigenomic breakdown leads to activation of damaging pathways, which likely further destabilizes the epigenome.
Okay, so we have specific players. Are there master controllers overseeing these processes, like the conductors of this whole genetic orchestra?
Yes, they looked upstream as well. The study highlighted something quite interesting, maybe a bit counterintuitive, about the genes linked to epigenomic erosion. These erosion-linked genes were enriched for components related to the polycomb repressive complex, PRC1, and PRC2, and other heterochromatin stabilizers.
Wait, polycomb complexes usually repress genes, keep things quiet, right?
Exactly. Think of them as the brain's internal silencers for certain genes, crucial for maintaining cell identity by keeping inappropriate genes off. So the findings suggest that it's the loss of this polycomb-mediated repression, when these crucial silencers start to fail, that actually contributes to the epigenomic erosion. It implies that maintaining proper polycomb silencing activity is actually protective against the disease's effects on the epigenome.
That's fascinating. Losing the off switches causes the chaos.
It seems to be a key part of the story, and it all ties back beautifully to the bigger picture they painted. The overall epigenomic information level measured in a brain sample directly correlates with better cognitive function and resilience. And just as importantly, it negatively correlates with all the classic hallmarks of AD pathology: the amount of amyloid plaques, tau tangles, neuroinflammation, higher information, less pathology, better cognition. It really emerges as a fundamental measure of brain health in the context of Alzheimer's.
This study, then, it really isn't just presenting a massive data set. It feels like it's fundamentally shifting how we should think about Alzheimer's. What makes this particular research so innovative compared to what came before? How does it open doors for future work?
Yeah, it introduces several really key innovations that push the field forward significantly. Firstly, as we touched on, it goes beyond the single region analysis. So many previous studies, valuable as they were, focused on just one brain area, often the prefrontal cortex. This study looked at 6 regions simultaneously.
Allowing direct comparison.
Exactly. That allowed them to make direct comparisons and reveal these crucial region-specific vulnerabilities, but also region-specific resilience mechanisms. It helps reframe Alzheimer's. Maybe it's not just one monolithic disease, but more like a coordinated regulatory disorder whose trajectories differ across brain circuits.
More nuanced view.
A much more nuanced view.
Yeah.
Secondly, that multi-omic integration we discussed is groundbreaking. Getting both the epigenomic, ATAC-seq, and transcriptomic RNA-seq data from the very same single cells is technically challenging, but incredibly powerful. It lets you directly link changes in chromatin accessibility, the switches, to the resulting changes in gene expression. That gives a much deeper, more mechanistic understanding of the regulatory programs involved.
And modeling.
Right. Thirdly, they introduced sophisticated trajectory and stability modeling. They weren't just looking for lists of genes that go up or down. They developed methods to quantify this systems-level drift in regulatory identity as the disease progresses, capturing the broader, more fundamental disruption of what makes a cell type distinct.
Measuring the loss of identity itself.
In a way, yes. And perhaps most excitingly, fourth, is their explicit focus on resilience. By specifically studying those individuals who maintain cognition despite pathology, they could identify these protective chromatin modules, the specific epigenomic patterns associated with fighting back against AD.
Shifting from just pathology to protection.
Exactly. That's a crucial shift in perspective, moving towards understanding what keeps brains healthy, not just what goes wrong.
It really does sound like this atlas is more than just data. It's practically a roadmap. So what are the practical implications? Does this research suggest new targets or strategies for actually treating Alzheimer's?
It absolutely functions as a roadmap. This incredibly comprehensive atlas provides potential mechanistic targets. We now have specific pathways, specific regulatory elements that are clearly either perturbed in AD or, importantly, preserved or even enhanced in resilience. By anchoring resilience in these concrete regulatory patterns, the study offers, as they put it, testable levers for intervention. We can start thinking about therapies aimed at specific molecular pathway.
Like trying to boost the protective patterns.
Precisely, or preventing the erosion. Imagine designing therapies aimed at restoring those protective chromatin configurations identified in resilient individuals, or finding ways to prevent that detrimental epigenomic erosion, maybe by supporting polycomb function, for example. It also offers potential for better stratification for clinical trials. Maybe we can identify patients based on their epigenomic profiles. This moves the field away from just describing the heterogeneity of the disease towards designing interventions with much greater therapeutic specificity. That's incredibly promising for developing more effective future treatments.
So this deep dive has really shown us that Alzheimer's, well, it's profoundly complex. It's not just about the amyloid plaques and tau tangles we hear so much about. It seems to be about a fundamental breakdown in our brain cells' own instruction manuals, their epigenome.
Yeah, a loss of identity at the cellular level. A fading of the very blueprint that keeps our cells healthy, specialized, and ultimately keeps our minds sharp.
But the really powerful insight here, the hopeful part, is that for those individuals with cognitive resilience, that epigenomic stability, that core identity, is remarkably preserved, even enhanced.
Absolutely. And that strongly suggests that perhaps the battle against Alzheimer's isn't only about clearing away the pathological junk, the plaques and tangles. Maybe it's just as much, or even more, about actively reinforcing the very foundation of cellular identity and function, stabilizing that epigenome.
So here's something to think about as you go about your day. If maintaining this epigenomic stability is indeed a key to cognitive resilience, what might that mean for a future where we could perhaps find ways to tune our epigenome to help our brains stay sharp, more resilient, even when faced with the challenges of aging or disease?
It's a really provocative thought, isn't it?
It really is. The future of Alzheimer's research might just involve learning how to rewrite or at least reinforce the script of cellular identity itself.